everyone, so today we are going to do paper October-November 2025 variant 1-3. There are multiple MCQs, multiple choice questions over here that we are going to look into one by one. Starting with the first one, we'll try and learn the way to solve these questions in less than a minute, so we can cater all 40 questions in due time of 1 hour 15 minutes. So starting with the first one, it says the diagram shows a stage micrometer, which is this stage micrometer, with divisions 0.1 mm, which is basically they are referring to this area. This is 0.1 mm viewed through an eyepiece containing a graticule. That's your eyepiece graticule that you can see. The same eyepiece is now used to examine a blood smear. How many graticule divisions will cover the diameter of a lymphocyte of 10 micrometer? First of all, in any of such questions, what you need to do is you need to do the calibration. What we can see over here is one stage micrometer, which is this. One stage micrometer is equal to 40 eyepiece graticule. So we can start with that right from 0.1 mm, which is one stage micrometer division, is equal to 40 eyepiece graticules. Now it's as simple as it looks. Now we simply have to calculate the value of one eyepiece graticule. So divided by 40 on either side, that is going to give you 0.1 divided by 40, that will be 0.0025 millimeter. Now as you can see, we have to measure the graticule division with respect to 10 micrometer. So let's convert it into micrometer. So simply multiply it with 1000 and it will be 2.5 micrometer. Now that is the width of one eyepiece graticule, right? Now it says how many graticule divisions will cover the diameter of a lymphocyte which has a diameter of 10 micrometer. So simple, you simply divide 10 by 2.5, which is the width of 1 micrometer. So that is going to give you 4. So 4 should be your answer. Moving on, in the second question, cell structures can be isolated by centrifuging the cells in a sucrose density gradient. This breaks up the cell and separates the cell structures into fractions. The diagram shows the position of some of these fractions in a sucrose density gradient. Now we can see over here that there are 4 fractions, and these fractions are always like this, in such a way that the more heavier it is, the more downward it is, right, in the centrifuge machine. The more lighter it is, the more upward it is, which we can see it over here. The ribosome is at the top, chloroplast is at the bottom of it. Now the question is, which fractions contain cell structure that carry out protein synthesis? Now we know for sure that ribosome is responsible for photosynthesis, but what else we need to know is that mitochondria and chloroplast both have plasmids, right? And if they have plasmids and they have 70S ribosomes, as we know, so with 70S ribosomes they can also synthesize protein. So there are 3 organelles over here, 1, 3 and 4, which can synthesize protein. So B should be our answer. Lysosome does not have any ribosome in it, so definitely it cannot synthesize protein. Moving on, the third question says the diagram shows a plant cell which labeled organelles would contain nucleotides. Now as we know, nucleotide is the monomer of nucleic acid, which can be DNA or RNA, right? Now keeping in mind DNA and RNA, we know that we can find DNA or plasmid in chloroplasts, we can find plasmids in mitochondria, and we can also find DNA or RNA in our ribosome. As we know, ribosome is made up of proteins and rRNA. So once again, rRNA is basically nucleic acid. So 1, 3 and 4 are the relevant answer. 2 is Golgi, and Golgi does not have any nucleotides. Moving on, the next question says the image shows is produced using a microscope. What we can see is basically a chloroplast image in detail. How many statements about this image are correct? The first one says it is an electron micrograph. Of course, the detail that you can see in this question is, in this image, is definitely showing that it's an electron micrograph. You can't see this kind of detail with a light microscope. Second, it shows part of a eukaryotic cell. So of course, chloroplast belongs to plant cells, and plant cells are eukaryotic cells. So this is also right. Third, it shows at least one mitochondrion, which is literally wrong. Why? Because you cannot find any mitochondria inside a chloroplast. So definitely it is wrong. Fourth one says it shows a specimen viewed at more than 400 times magnification. Of course, because 400 times magnification is used only in light microscope, and we have already said that it's an electron micrograph, so it must be much more than that. So this is also right. So there are 3 statements which are correct, so the answer should be C. Moving on, the next one says bacterial ribosome consists of 2 subunits and a total of 3 rRNA molecule per ribosome. Now the important point is to understand this. Okay, 3 rRNA molecule per ribosome. So one ribosome has 3 rRNA, remember? Now, an analysis of all the 70S ribosomes from a single cell of bacteria showed that there were 57,000 rRNA molecule. So you can even solve it from the first hint, but let's move on. The second one says 3 types of rRNA molecule, that we already know, and 19,000 copies of each type of rRNA molecule. And then it says how many 70S ribosomes were there in the bacterial cell. So we have already read 3 rRNA per ribosome, and here it already said 57,000 rRNA. So simply 57,000 divided by 3, that gives you 19,000. So it's B should be your answer, right? It means there are 19,000 ribosomes in total. And you can also double check it with the rest of the options, but you can simply solve it with the first one. Moving on, radioactively labeled amino acids are introduced into tracheal cell that uses them to make mucus, a glycoprotein. Which route will the amino acid take? Now we can see in this cell they have labeled a few things. Basically we have to start, we have to understand from where it is going to start. So the very first place where amino acid should reach in a cell is 1. What is 1? It's cytoplasm. So when a cell absorbs amino acid through diffusion or active transport, it directly comes into the cytoplasm. So 1, then followed by it goes into ribosome during the process of translation, that is protein synthesis, right? So amino acid join together to form a polypeptide chain. So that is the second step. After that it goes into the Golgi, which is 3, right? Why Golgi? To modify it. And finally it is packaged inside a vesicle, which is 2, right? So from 1 to 4, then 4 to 3, and then 3 to 2. So the relevant answer here is B. Alright, next one says which feature shown in the diagram can be present in viruses, right? So it's pretty much clear. 70S ribosomes are absent in viruses. DNA can be there, RNA can be there, and protein coat can be there. So these 3 things, 1, 2 and 4, can be there. So B should be your answer. That's pretty easy. Alright, then it says the diagram shows some steps in a laboratory method used to identify the presence of non-reducing sugar in a food sample. Okay, some terms are missing, step X, then step Y, and then the end color Z. Which terms are correct for the expected results for a low concentration? Okay, so low concentration here should be marked, very low concentration, right, of non-reducing sugar. So now first of all we have to see what should be step X and then what should be step Y. Of course step Y has to be Benedict test, because without that you can't do that. But before Benedict test, what you need to do is you need to do acid hydrolysis. Now with acid hydrolysis, so we have come to the conclusion that it's either A or C. Then you're going to look at the color change. So if you're going to look at the color change, here the end color should be, according to very low concentration, it should be green and not orange, because orange means moderate concentration. So C should be our answer. Alright, Then many biological molecules exist as polymers made of monomer subunits. Which row correctly identifies the monomer in each of the biological molecule? It's simply AO1 concept, which means simply replication of knowledge. What is collagen made up of? Definitely amino acid, so it's either A or D. Then what is glycogen made up of? It has to be made up of beta glucose or alpha glucose. No, alpha glucose, of course. And then cellulose is made up of beta glucose. So what is relevant is A over here. All right. Moving on to question number 10. Which statements correctly explain the results when the two molecules shown in the diagram react together? So what we can see over here are two molecules of glucose which are reacting together in the presence of water, right? So which particular statement is more relevant over here? So what looks like, okay, so when we add water, what are we doing? Hydrolysis, of course. So we want to break down this disaccharide into two monosaccharide, which is what B says. A reducing sugar is split. And why it is a reducing sugar? Because it is the structure of maltose. Okay, so maltose is a reducing sugar. It's split into two monosaccharides, which are alpha glucose by hydrolysis. So B is relevant over here. 11. Which row describes amylopectin? So amylopectin is branched, as we know. So that is the first thing that comes to my mind, right? So according to that, it can be either A or C. And then, of course, if it is branched, it must have both alpha 1,4 and 1,6. So that goes with that. And then the function of the amylopectin is structural or storage. So its main function is relevant to storage. Why? Because amylopectin along with amylose makes starch, and starch's job is to store carbs, right? So that's A should be our answer. Moving on to 12. What is a feature of triglyceride molecule? They are hydrophobic molecule. That's it. That's the simple most answer. You can look into it. Definitely, as a good student, you have to look at the rest of the options in order to make sure that you're choosing the most correct answer. So B says with single ester bond, so it has 3 ester bonds. C says it are polar molecules. They are not polar, of course, and of course D, they are not water soluble. So that cannot be used. All right, moving on to an unsaturated fatty acid molecule has 17 carbon atoms in the tail, which is attached to its carboxyl group. The tail contains 3 double bonds. How many hydrogen atoms does the tail contain? Now, this is more like a mathematical question. I think one should do it like practically over here. How many carbons are there in the tail? 17. So let's make 17 carbons. That is going to make it much more easier for us. 1, 2, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 and 17. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Okay, and then a carboxyl group is also attached to it. So let's make another carboxyl group. Okay, and then it says it has 3 double bonds. So let's make 3 double bonds anywhere in the chain. 1, 2 and 3. And now let's add carbons. 1, 2, 1, 2, 3, 1, 1, 2, 1, 2, 1, 2, 1, 1, 2, 3, 4. Just make sure that every carbon should be satisfied with 4 bonds. Okay, so you should not make more than 4 at any point. 1, 1. Okay, so I made all of them. Now we can count them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29. So A is our answer. Okay, so for some questions you cannot simply mathematically calculate it. Actually, you can calculate it, but then you are not sure, because it's a good educated guess, but you're not sure about it. So the better option would be that you do it mathematically, you solve it right there. All right, next, which statement about phospholipids in the bilayer of a cell surface membrane is correct? This is a pretty easy question. The heads should be facing Okay, first of all, the heads are not non-polar, so that's wrong, right? Then the polar phosphate heads face outward and the non-polar fatty acid tail face inward. That's the correct answer. That's how you always define the structure of phospholipid. All right, question number 15 then. Which description of enzyme that use the lock and key hypothesis are correct? So in lock and key hypothesis, the active site is always complementary to the substrate that the enzyme acts on. That's perfectly all right. Second, it says bonds that form in the enzyme substrate complex change the shape of the active site. No, it doesn't. So that's the role of an inhibitor, which is not involved over here. Third says most of the amino acid in an enzyme help to maintain the specific shape of the enzyme. Of course, amino acid in an enzyme structure provide the primary structure of protein, and I believe you should remember that if you change the primary structure, it is going to change the tertiary structure. So that's why the specific shape is going to be changed. So it's 1 and 3.